So we know every nucleated cell in our body has DNA. And we know this DNA holds information based on the sequence of these base pairs. And this specific information can be used to create a specific sequence of RNA molecules. And now this RNA molecule holds information. And now based on this very specific RNA sequence, we can use it to create a very specific protein. And we know these proteins are incredibly important. They regulate all the chemical reactions that occur in our body. And something important to realize is DNA is just another molecule. And like any other molecule, it can be chemically modified and damaged. For example, these nucleobases, they can be arylaminated or alkylated or oxidized. So when these nucleobases get chemically modified, they become damaged. And once we damage this DNA molecule, now that information is lost. So once we damage our DNA, that information is lost, and now we'll create the wrong RNA, which will then create wrong proteins. And now these proteins will not regulate the chemical reactions that occur in our body, and this can lead to cancer and cellular senescence and inflammation. So this is bad. So it's very important to maintain the integrity of our DNA molecules in our nuclei. So there are different ways to characterize DNA mutations, but one way is to look at transition mutations and transversion mutations. So if you have a purine that gets chemically mutated into another purine, or if you have a pyrimidine that gets chemically mutated into another pyrimidine, that's a transition mutation. However, if you have a purine that gets chemically modified into a pyrimidine, or if you have a pyrimidine that gets chemically modified into a purine, that's called a transversion mutation. However, we can also have point mutations. For example, let's say this is the normal DNA sequence, which encodes for this normal mRNA, which we know is a codon that encodes for a lysine residue. However, let's say we have a DNA mutation where instead of a C, we chemically modify it into a T. What would happen? Well, now we would encode for a new mRNA codon. However, this mRNA codon also codes for a lysine. So even though we had a DNA mutation, we still encoded for a lysine, so this was a silent mutation. However, let's say we took this T and we chemically modified it, we went through a DNA mutation where now it's converted into an A. Well, now it would encode for this stop codon, which will suddenly cease the production of this polypeptide, so we call this a nonsense mutation. However, let's say we chemically modify this T and mutate it into a C, well, now we would encode for this codon, which encodes for arginine. So something important to realize is lysine is positively charged, and so is arginine. So even though we went through a chemical mutation which created a new amino acid, this amino acid is very similar to lysine. They have very similar chemical properties, so this is a conservative missense mutation. However, let's say we went through another DNA mutation where now we encoded for this threonine amino acid. Well, threonine has very different chemical properties relative to lysine. So this is called a non-conservative missense mutation. So there are two types of missense mutations. If we have a DNA mutation that creates a similar amino acid, that's conservative. If we go through a DNA mutation that creates a very different amino acid, that's a non-conservative missense point mutation. So now that we've learned about the different types of DNA damage, now let's talk about the different types of DNA repair. But something important to realize, I've artificially characterized these different forms of DNA repair. So this is a bit arbitrary. And to the cell, there's just DNA repair. So these are artificial ways to characterize the different types of DNA repair. And there's overlap in these mechanisms. For example, a type of DNA damage are thymidine dimers. However, to the cell, there are different ways to fix these thymidine dimers. For example, we can go through direct reversal repair, or we can go through nucleotide excision repair. So there's overlap in these mechanisms, so just keep that in mind. So the first type of DNA repair I want to talk about is direct reversal, where we directly reverse DNA damage. For example, let's say we have two thymine nucleobases next to each other. What can happen is they can absorb energy in the form of UV light and use the energy in those photons to form covalent bonds with each other. And once they form covalent bonds, now we've damaged this DNA molecule. We've chemically modified and damaged it. However, fortunately, we have this enzyme, this MTHF enzyme, which can absorb energy from blue light. In this blue light, there are photons with energy. So this MTHF enzyme can essentially absorb energy from blue light and energize electrons, put electrons in higher energy states, and then donate those electrons to FAD, which gains those high energy electrons and uses those high energy electrons to break those covalent bonds and fixing the DNA molecule. So in this example, we've directly reversed this DNA damage, and this is referred to as photolyase. However, there are other types of direct reverse DNA repair. And something really important to realize is, again, DNA is just a molecule that's made out of lots of nucleobases. And these nucleobases can be very nucleophilic. So therefore, if we ingest electrophiles, our DNA, which is nucleophilic, can react with these electrophiles, going through chemical reactions and therefore chemically modifying and damaging DNA. For example, this oxygen in this G nucleobase, this oxygen is very nucleophilic. So therefore, if we ingest an electrophile, for example, ethyl methanosulfonate, this particular carbon is very electrophilic. So therefore, we can go through a chemical reaction where this oxygen nucleophilically attacks this carbon, forming a bond which we see right here. So when we go through this chemical reaction, we chemically modify this G base. So now it's chemically modified and damaged. So something important to realize is normally when we replicate this DNA, we know G will base pair with C, and then we'll create a normal replicated genome. However, when we go through this chemical modification where this G goes through the nucleophilic reaction and gets chemically modified, now it's chemically different. So now it's when we go through DNA replication, it's going to base pair with the T. So if it base pairs with the T, we've created this mutated replicated genome. So that's bad. This mutated replicated genome will now encode for damaged proteins. So this is bad. This DNA mutation is very bad and dangerous because now the information is lost, which will now create faulty proteins. So we can see it's very dangerous for this nucleobase to be chemically modified and mutated. However, fortunately, we have this MGMT enzyme. And this MGMT enzyme has a very nucleophilic sulfur. So this sulfur can essentially nucleophilically attack this carbon, leading to this bond breaking and these electrons falling on this oxygen. And the mechanism's not really important. The point is this MGMT enzyme can essentially snatch this alkyl group. It can snatch and take away this alkyl group. So in the process, this MGMT enzyme gets alkylated. It snatches that alkyl group, so it gets alkylated. However, in that process, we fix this nucleobase. This nucleobase is fixed. We've directly reversed that DNA damage. So this is another form of direct reversal, DNA repair, where we essentially directly reverse that damage, and in that process, we repaired this nucleobase. So now the nucleobase is back to normal. So the second type of DNA repair I want to talk about is mismatch repair. So we know when we replicate DNA, we have this DNA polymerase, which uses this template strand to add new nucleobases to replicate this DNA. However, this DNA polymerase is not perfect, and every now and then it accidentally adds the wrong nucleobase. For example, if we have the C nucleobase, we should add a G nucleobase. However, every now and then the DNA polymerase makes a mistake and instead adds an A nucleobase, which is bad. That's the wrong pair. But fortunately, this DNA polymerase has this mismatch repair mechanism, which it senses we have a mismatch, so it chops off and releases this A, and then adds the right nucleobase. So then it will add this G, the correct nucleobase. So this is mismatch repair. However, the DNA polymerase is not perfect, and every now and then it's going to accidentally miss one of these mismatch pairs. It's going to miss it and it's going to leave it here. So now we have this mismatch. Now we have this A when we really should have a G. So we're going to have this mismatch bond, which is going to cause a physical distortion. It's going to cause a physical bulge in this DNA strand. And now we have this wrong strand. However, fortunately, our cells have this trick, this third type of DNA repair called nucleotide excision repair, which is this trick the cell uses. And we're going to see this mechanism over and over again. So this trick that the cells use is, first, we use an endonuclease. And this endonuclease essentially causes a nick in the phosphodiester backbone, essentially breaking these covalent bonds, which then allows the endonuclease to take this damaged region and to rip it off, creating this empty region. Now that we have this empty region, now DNA polymerase can come along and add the right nucleobases. So now we can add the right nucleobases. We can add a G, the right nucleobases. And once we add the right nucleobases, then we have this DNA ligase, which ligates the phosphodiester backbone, these covalent linkages, and now we fix the DNA. We repaired the DNA. So these three steps are referred to as nucleotide excision repair. And this is a trick. These three steps is a trick the cell uses to broadly fix different types of DNA damage. And we're going to see this mechanism over and over again, this nucleotide excision repair, these three-step trick. So again, this nucleotide excision repair is a very powerful trick the cell can use to fix many different types of DNA damage. For example, maybe we have this DNA damage. Maybe we have a mismatch pair, or we just have this general DNA damage. We can fix this using nucleotide excision repair. So remember, the first step is to use this endonuclease to break this covalent backbone. So the point is, this endonuclease is a nucleophile which reacts with water, making water more nucleophilic, which attacks this electrophilic phosphate. The organic chemistry mechanism isn't really important, but the point is this endonuclease breaks this covalent bond and therefore breaking the backbone of DNA. So we would do that here and we would do that here. So now that we've broken this backbone, these covalent bonds, now we can remove this entire stretch of DNA. And remember, here's where the DNA damage is. So if we remove this entire stretch of DNA, we remove the DNA damage. So once we remove this entire stretch, now we have this empty region. So now DNA polymerase can come along, adding the correct nucleotides, adding the correct nucleobases. Then the next step is ligase comes along, which essentially ligates this covalent backbone, the DNA backbone. So once this DNA ligase ligates this DNA backbone, now we fix the DNA. Now we have an intact, fixed double strand with the right nucleobase. So this nucleotide excision repair, these three steps, is a very powerful and broad technique the cell uses to fix many different types of DNA damage. We can also use this nucleotide excision repair trick to fix these thymidine dimers. Remember, when we have two thymine nucleobases that absorb energy from UV light, we can form these covalent bonds forming this thymidine dimer. And this thymidine dimer is chemically damaged DNA, which causes distortion in the DNA strand. We essentially cause this bulge and we cause this distortion in the DNA strand. So these nucleotide excision repair mechanism and these enzymes sense this distorted DNA strand, this physical distortion. So then we can use our nucleotide excision repair trick to fix this. Again, remember, first step, endonuclease breaks the backbone. Then we remove this damaged DNA. Then DNA polymerase comes along, adding the correct DNA. Then we use a DNA ligase to ligate the bonds, fixing the DNA. So again, the point is this nucleotide excision repair is a broad way to repair different types of DNA damage with these same three steps. Another type of DNA damage is through these polycyclic aromatic hydrocarbons, which is a carcinogen. So this polycyclic aromatic hydrocarbon on its own is fine. However, when it enters the liver, it gets chemically modified into this epoxide form. And this epoxide is very electrophilic. So therefore, because this epoxide form is so electrophilic, it can react with DNA molecules and these nucleobases, which are nucleophilic. So this nucleophilic nitrogen can nucleophilically attack this carbon in this epoxide, essentially forming a covalent bond. So we chemically modify this DNA nucleobase. So now we've damaged the DNA. We've added this polycyclic aromatic hydrocarbon, so we've chemically modified and damaged the DNA. So now that we've added this polycyclic aromatic hydrocarbon to this nucleobase, we've chemically modified and damaged this DNA. We've added this huge, bulky alkyl group. So this is very bulky. So this is going to cause a physical distortion in our DNA strand because we've added this huge, bulky polycyclic aromatic hydrocarbon. However, fortunately, this nucleotide excision repair trick, this pathway, senses this bulky distortion, this physically distorted DNA strand. So we can fix it using this nucleotide excision repair trick. And remember, the first step is the endonuclease nicks this phosphodiester backbone, breaking this covalent bond, which then can remove this entire stretch of DNA, creating this free open region. And now DNA polymerase can come along, adding the correct nucleobases, and then ligase can come along and ligase the covalent bond, fixing and linking the covalent bond, fixing the DNA. So again, you can see now how this nucleotide excision repair is a very broad way to fix lots of different types of DNA damage. And something important to realize is this nucleotide excision repair pathway explicitly senses and searches for these physical distortions, these physical distortions to the DNA backbone. And they sense these physical distortions and then activate this pathway to essentially remove this physical distortion and to fix it and add new correct DNA to fix the DNA strand. Another type of DNA repair is base excision repair, which is very similar to nucleotide excision repair. So the key idea of base excision repair is sometimes we chemically modify and damage these nucleobases. For example, we know our mitochondria and our electron transport chain is constantly going through oxidative stress, creating free radicals. And we know these free radicals are very chemically reactive. So they can chemically react with, modify, and damage these nucleobases. For example, this is normally what this G nucleobase looks like. However, when these free radicals react with this G nucleobase, we can chemically modify to look like this, where we chemically modify, we oxidate it. And now that we've chemically modified it, now it's going to distort. It's going to distort, and now it's not going to base pair correctly because we know normally G base pairs with C. However, when we oxidate this G nucleobase, now it's chemically modified. So now instead of base pairing with C, now instead it's going to base pair with A. For example, when we replicate this genome, we know we open up the double strand. And once we open up the double strand, now we use this as a template to add a new DNA strand. However, because we've oxidated this G and we chemically modified this G nucleobase, now instead of adding a C, it's going to add an A. We're going to have this mismatch pair. So this is bad because now this new strand of DNA has the wrong information. It should have a C, but now it has an A. So this is bad. So this damaged base is very bad, so we need to get rid of it. But fortunately, we can get rid of it through this base excision repair. So we have these glycosylase enzymes that sense this damaged nucleobase. So essentially, this glycosylase enzyme can essentially hydrolyze this N-glycosidic linkage. And when it hydrolyzes this N-glycosidic linkage, it breaks this covalent bond, which can release this damaged nucleobase. So now we release this damaged nucleobase, but now we have this empty region. And we call this empty region an AP site, an A-basic site, a site without a base. So now we have this empty region, so that's not good. But fortunately, we have these AP endonucleases that sense this empty region. So once these AP endonucleases sense this empty region, now we can go through a very similar pathway as nucleotide excision repair. Those same 3 steps that we learned earlier. We can go through a very similar mechanism where this AP endonuclease breaks this backbone, breaks these covalent bonds and breaks this backbone. And once we break these covalent bonds and break this backbone, now we can essentially remove this stretch of DNA, removing this abasic site. So now when we remove this stretch of DNA, now we go through the same steps. Then DNA polymerase comes along, adds the correct nucleobases, adds a correct non-damaged G nucleobase. Then ligase comes along, and ligase is the backbone. And now we fixed the DNA damage. Now we fixed, and now we have this normal DNA strand. So this is base excision repair, which is a very similar mechanism. However, there are other circumstances where we need to go through base excision repair. For example, every now and then we go through spontaneous depurination events, where these purine nucleobases spontaneously pop off. Once they do that, we're left with this abasic site, so that's a problem. Also, every now and then we go through spontaneous deamination events, where these nucleobases, this amine group, pops off. We go through deamination events where we replace this amine group with the carbonyl group. And this is bad because now that we've chemically modified this cytosine nucleobase, now it's going to mismatch incorrectly, because we know usually cytosine matches with guanine. However, when we go through that spontaneous deamination event, now it's going to match with adenine. So that's going to induce mutations. However, fortunately, we have these glycosylase enzymes that sense these chemically modified nucleobases. And essentially what these glycosylase enzymes do is they sense these chemically modified nucleobases, and then they chop off this bond, releasing this damaged nucleobase. And once we do that, we're left with this abasic site. So we can see how we can be left with these abasic sites, and that's a problem. So we need to fix this. So the way we fix this is similar to the way we go through nucleotide excision repair. For example, the first step is we have one of those endonucleases, in this situation an AP endonuclease, that senses these AP sites. And we know what these endonucleases do. They essentially chop these phosphodiester linkages. They chop off, they break these covalent bonds, essentially releasing this entire region of damaged DNA. And once they do that, now we know the next step. Now we know DNA polymerase comes along and adds the correct nucleobases. Once we do that, then DNA ligase comes along, and we know we ligase the backbone, forming these covalent bonds, and now we fixed the DNA. So again, we see base excision repair is very similar to nucleotide excision repair. The only difference is we had that AP endonuclease that senses those AP sites, those AP sites. So the last type of DNA repair I want to talk about is homologous and non-homologous DNA repair. So our DNA faces lots of different types of environmental insults. For example, we know our mitochondria is constantly producing these reactive oxygen species, and also we know the cell can be exposed to high energy radiation. For example, high energy photons from X-rays and UV light. So these environmental insults can damage DNA. And in fact, they can break these covalent bonds in this phosphodiester backbone, leading to a single strand break. Because again, we know DNA is made out of two strands. So sometimes we can damage one strand and essentially break this covalent bond, leading to a single strand break because one strand has been broken. However, usually this is pretty easy to fix. We can just ligase this back together. Now we fixed it. However, every now and then we have a double strand break where we break both strands. Now this is very dangerous because once we break both strands, now this DNA strand is not connected to each other. So essentially, once we break both strands, now we can go through different types of DNA damage. For example, genome rearrangements and compromised genome integrity, which can lead to cell death. So there are two mechanisms the cell uses to fix double strand breaks. There's non-homologous DNA repair and homologous DNA repair. So first, let's talk about non-homologous DNA repair. So in non-homologous DNA repair, we know the first step is to cause a double strand break. So we go through some kind of environmental insult where we cause a double strand break, which we know is very dangerous. This can lead to genome rearrangements and cell death. So we need to fix this double strand break. So the first step is enzymes come along and essentially shave off excess nucleobases. So we essentially shave off excess nucleobases, which we lose, to cause a clean break. So now we have a clean cut. So now once we have a clean cut, other enzymes can come along and bind to both of these ends. And once we bind to both of these ends, now these enzymes can essentially seal this double strand shut. It can seal this double strand shut. And now we fixed the double strand break. So we had a double strand break. We shave off the edges, creating a clean break, which then enzymes can come along and essentially seal shut. And now we've repaired the DNA. However, is this DNA fully repaired? Well, not quite, because if you think about it, we've lost DNA information. Because remember, we shaved off excess nucleobases, so we lost information in these excess nucleobases, which were lost. So this entire region of the genome has been lost. That DNA information has been lost. And then we sealed it shut. But this is a problem. Again, we've lost very important DNA information. For example, maybe this was the entire gene, but now we've lost this entire region of the gene. So now we have a new damaged gene. So this is a problem. So why would the cell do this? Well, if we didn't fix this double strand break, the cell would die. So this is the best case scenario once we've caused this break. So this double strand break is very dangerous. And if we don't fix it, the cell will die. So what the cell does is it settles to snap shut this double strand break, and in that process we lose DNA information. So that's a problem. So this non-homologous DNA repair isn't an ideal situation. So now let's talk about homologous DNA repair, which is a much better mechanism for fixing double strand breaks. So the key point to realize about homologous DNA repair is to realize early on in the cell cycle, we have one copy of each chromosome. For example, let's say we're talking about our chromosome 1 that we get from our mom. We have one copy of this chromosome 1 that we get from our mother. However, after the S phase of the cell cycle, remember, we copy each strand of DNA. So now we have a sister chromatid of this chromosome 1 from our mother. So we essentially have this backup copy. We essentially have a backup copy of this chromosome 1 that we got from our mother. So we can use this backup copy because it has information. It has DNA information in this backup copy. So now let's say we have DNA damage where we cause a double strand break. So this may be a problem because we've caused this double strand break, and you can say that we've lost DNA information. However, keep in mind we have the sister chromatid. We have a backup copy. So the information is saved in this backup copy, in this sister chromatid. So now we can fix this double strand break using the information in our sister chromatid. So the first step is these enzymes come along and essentially they shave off single strands of this double strand. So they'll shave off single strands of this double strand to create these empty regions. And now a new enzyme will come along and will essentially take this strand of DNA and essentially use it to bind to our backup copy so we can use the information in this backup copy as a template to add on new DNA, the right information. So first we would open up this backup copy so we can access it. And now this enzyme is going to take this strand and essentially bring it over, and we use this strand as a template because this strand has DNA information. So we bind and we use this backup copy as a template. So now that we use it as a template, we can add the correct DNA information. So now we've added all the correct DNA information. Then this strand will come back. Now it'll come back, and now that it's come back, we have the right DNA information. So we can use this as a template to add this new strand. So now another enzyme will come along and we'll add this new strand. Essentially a polymerase will come along and add this new strand. So now once we've done this, now the last step is to ligate these two edges. So ligase will come along and ligate these two edges. And now we've fully repaired this double strand break. And this is good information. This is good. We fully repaired this double-strand break. And the key idea is when we cause this double-strand break, the key idea is with homologous DNA repair, we use the homologous chromosome, which had the DNA information. So that information was saved. So we could use that information in the homologous chromosome to fix this double-strand break. And now we fully recovered and fixed this double-strand break, which has good information. So this homologous DNA repair can only occur once we've gone through the S phase, because only once we've gone through the S phase do we have this backup copy. But as long as we've gone through the S phase and have this backup copy, we can go through homologous DNA repair, where we fix the double-strand break and we fully fix this double-strand break. So hopefully by now you can see how there are many different sources of DNA damage that can lead to many different types of DNA damage, which can be fixed through many different types of DNA repair. However, there seem to be two major sources of DNA damage: high energy electromagnetic radiation, for example, X-rays and UV light. These high energy photons can essentially rip off electrons and break in covalent bonds, damaging the DNA molecule. And the other source of damage is the inflammation. Inflammation creates free radicals and oxygen radicals, which are also very reactive molecules which react with the DNA molecule, chemically modifying and damaging the DNA molecule. However, fortunately, there are many different types of DNA repair. And these are a lot of the enzymes that are involved in DNA repair. So making sure you have high expression and activation of these different enzymes and these different genes that fix DNA damage is very important for cellular health.